My fellow Americans, the following footage may shock and disturb you.
We have reports that a giant evil hamster is taking over New York City.
To combat this issue, we have called upon our scientific community to find a solution.
We have a solution.
Let me explain.
Here's our plan. The solution we have come up with uses SCID, also known as severe combined immunodeficiency. Patients with SCID lack a functional immune system and are very susceptible to harmful pathogens. If not treated immediately and constantly, SCID is very deadly. The NIH lists this as a rare disease. 1 in 50,000 children are born with SCID in the United States annually. SCID is caused by the absence or malfunction of T cells. T cells are required to activate B cells, which produce antibodies and play a key role in the adaptive immune system. A famous case of SCID is David Veeder, also known as Bubble Boy. He was born with SCID and lived to the ripe old age of 12 when he died of Burkitt's lymphoma. The movie Bubble Boy has given us a comedic idea of what would be the life of a person with SCID. Alright, so we found a gene, interleukin-7. Interleukin-7 is important for T and B cell development. IL-7 stimulates proliferation of all cells involved in the adaptive immune response, and it's also important for proliferation during certain stages of T cell development, survival, and homeostasis. It works as a signaling molecule and forms a heterodimer between two cell surface receptors and promotes T cell development. We set out to see if by disrupting the binding of IL-7 to its receptors, IL-7 alpha and the common gamma C receptor, we could prevent T cell development and create the SCID phenotype.
We will perform our tests using an immunocompetent mouse model, which we believe will give us proof that our method will work against this monster. We will use an adeno-associated virus to infect mice with a CRISPR construct targeting the interleukin-7 gene in stromal cells. The CRISPR-Cas9 system has been shown to be successful in correcting disease mutations in adult mice, but we are hoping to knock out IL-7 in enough cells to induce a disease mutation, which, if successful, will be a first. After the virus injects the construct into the mouse's cells, the construct will integrate into the genome and begin to be expressed. This construct should knock out the IL-7 gene, which we hypothesize will inhibit T cell development and induce the SCID phenotype. This will be verified through Western blotting. If we can successfully produce the SCID phenotype in adult mice by knocking out IL-7 in enough stromal cells, we will use an airborne rodent-specific adenovirus to infect the monster and put an end to this senseless destruction. By conducting this experiment, we hope to save our country, prove that IL-7 is crucial for the adaptive immune response in vertebrates, and show that the CRISPR-Cas9 system can be used to induce loss-of-function mutations in adult organisms.
Thanks to our brilliant scientists, the problem has been exterminated.